I must say, Danielle, that's very bloody tame. I was expecting a bit more, I don't know, spice in the intro. So anyway, so good on you. So look, I'm just so amazed. Welcome to Sydney, guys, my now adopted hometown. It's amazing to see what a single comment from Rob Bird has led to. Build a website, and really Danielle has built an amazing community and support group really for our patients, and it's really quite amazing to see how it's blossomed. So I'm very privileged to be a part of this. So thank you for having me here. So I'm going to be talking about something a little bit more, a bit of hope along the horizon. Now, it is a bit of a nerdy talk because I'm talking about really sexy new drugs that are unpronounceable, and to be honest, it's like a sobriety test for us, after a few drinks, I reckon. So, we're going to cover really what's very soon going to be available. So rilzabrutinib being one, and also other drugs that are currently in sort of clinical trials that, a lot of us in this room actually are currently enrolling patients in. So we can't talk about, I guess, these new fancy drugs without sort of understanding, I guess, the mechanism of ITP. And I know, most of you in the audience might be well kind of aware of this, but I just want to go through a top-line sort of rundown. So really, what is ITP? Well, we know that we're meant to have normal platelets. They're floating around like Band-Aids, plugging bleeding spots. Of course, if you're low on that, it's no good. Now, we know the cause of ITP is basically immune confusion at the heart of it all. Okay, so for whatever reason, something's happened, lightning has struck, the immune system mistakenly thinks the platelets are foreign and starts producing, at least some of the immune cells, start producing antibodies directing against our own platelets, which it shouldn't do. Now, those platelets basically are then tagged by these antibodies so that when they float into the spleen area, and I kind of like to think of the spleen as kind of the customs at the airport where they're trying to check your baggage, and like me, probably bought too much crap. And they're basically looking for stuff that's been tagged as foreign and then basically chomp them up and take them out of circulation. So you can imagine then, that's no good when all your platelets are being tagged by that cranky customs official to take out your platelets. So a little bit of background. So how the heck does the body know how to make platelets, right? It's actually quite an elegant system. So we know the liver produces the kind of hormone that tells the body or the marrow factory to make more platelets. So that hormone's called TPO. And I know, Rob has just mentioned, obviously one of the drugs we use, a TPO receptor agonist. So TPO is a cousin of EPO, the way to think about it, Lance Armstrong's favorite drug. And it basically tells the body to please make more platelets. So, and that's what it does. So you basically use the whip that cracks on to the marrow factory as they churn out, please, more bloody platelets. And we know quite elegantly that the TPO kind of gets absorbed into platelets. So kind of beautiful in that if you've got normal amounts of platelets, you'll suck up all the TPO, so the circulating levels are lower, because you don't need to make too much when you've got normal platelets. And when the platelets drop, there should be more TPO to tell the marrow factory to please make up and make more platelets. So that's what happens normally. Now, we know if everything's going according to plan, TPO will be, basically getting our marrow factory to churn out these platelets, to hang around for 5 to 9 days, and she'll be right. And we know, of course, then when they die, the spleen, customs again, actually go and recycle and kill off those dying platelets. But of course, is the circle of life that, the marrow will just keep producing more and more and more. But in ITP, something goes wrong. So interestingly, there's been observation that there seems to be lower levels of TPO in the blood of ITP patients. That then, of course, means that there's a production issue. The megakaryocytes are, or really the granddaddy cells in the bone marrow that sort of give birth to the platelets. And we think it's probably because the antibodies are attaching to them and stopping them from working properly or killing them. So it's not really making enough platelets. Of course, then the double whammy, though, is that, remember I mentioned about the immune confusion. So these platelets that have been sent out into the wild, hoping to plug bleeding spots, they're now being tagged by our own immune system to then be taken out. So we have reduced production, but also our immune system, very sadly, is tagging them to be destroyed. They don't hang around for very long, and therefore the platelets, of course, drop. And and this kind of makes sense now why once upon a time splenectomy, and I guess still now, but why splenectomy works in some patients, because we know that that's where they're being destroyed, right? So if we remove that site of destruction, so we just bomb customs, then they'll hang out for a bit more. I could bring in more overloaded luggage. So that's the whole, I guess, reason why we take out spleens once upon a time. So now this understanding, and the reason I want to do this is to set the scene, right? Because now that we know what happens and what goes wrong, very smart people means they could develop fancy new drugs. So we know, okay, not enough TPO. That's great. That's why we have TPO receptor agonists, right? So these 3 drugs are the ones that are available, here in Australia. We were able to basically increase the production then of platelets in our patients with ITP. Now, what about this particular arm? Okay, so there's antibodies that are being produced, and no surprise then, like the drugs that we're using are targeting the immune system to try to calm it down and stop it from making new antibodies. So steroids, as Daniel called them, Satan's Tic Tacs, that's what it does. It just suppresses the immune system, so you stop trying to recognize platelets as being foreign. Rituximab mycophenolate really does the similar sort of action. Now, and as I alluded to before, now we know, yes, spleen's the main site of production, so let's just, chop it out. And I'm sure the surgeons use a much more refined instrument in their operations. I like to think, although I'm not so sure sometimes, speaking to them. So, now, this is really the current state of play at the moment, right? So, that's for the Gucci comment, Rob, for the last meeting. That's pretty much current state of play. That we look upon clinical trials, and that really is the future. So before I sort of talk about these clinical trials, I'm sorry I've got to show this very complicated diagram, which is a more kind of advanced sort of version of what I've been trying to convey in the initial introduction slide. And the reason I want to show this is because now that we really understand what goes wrong in ITP down to the immune cell level, it means, again, very smart people have come up with ways that really is the next generation of ITP treatments. So I'm going to try to walk us through this ridiculous sort of convoluted diagram and kind of try to set the scene in terms of, well, why these new drugs are coming about, where are they sort of intervening in this sort of abnormal immune response. So firstly, I might try to go through this, because it is, I know, a little bit busy, but really the way to think about it is our immune system's designed where we have the scout, which is what's called the B cells, to sort of try to determine what's foreign to the body, right? So they have to go around and check every cell to say, are they friend or foe? And if they think something is not, part of our own body, it requires this buddy, what's called the T cell, to say, hey mate, is that thing dodgy? And the T cell's got to go, yep, that looks dodgy. So they kind of do a handshake, literally. They sort of interact with each other. And there's another actually, cell that's called a macrophage. And in the macrophage, I guess, what's the word? It's probably the bouncer in your nightclub that picks out dodgy people too, right? It kind of tells them, hey, that person looks a bit dodgy. He's way too drunk. It's Danny Hsu. Don't let him in. So they're the ones that are actually presenting, what we think is foreign through the platelets in ITP to the B cell, the scout, and then the T cell, its buddy, will say, yeah, look, that platelet does look a bit dodgy. So one of the drugs that have been, developed is rilzabrutinib, and it belongs to this class of drugs called BTK inhibitors. Yeah, I'm sorry for the ridiculous number of acronyms we use. So rilzabrutinib, so there you go, you can take a picture. Don't just spell it, because I can't, like Rob. Or Rob can spell, I can't. Now, it's really quite elegant. What it basically does, it blocks the destruction of your tagged platelets, right? So on that right-hand side, and I must say, if you're over 40, like I am, you're not going to be able to read that from the back. But what it basically is able to do is block these tagged platelets from being chomped up by our cells in the spleen. And also, very importantly, it reduces the production of antibodies by its effect on the B cells, the scouts. Okay. Very importantly, it's got no effect on how the platelets function. And the reason I mention that is these class of drugs, we've been using them for actually many, many years in sort of the leukemia world. And it was the observation, in fact, that in those trials that, well, some of those patients have increased platelet counts, which led to the development of this class of drugs for ITP. So really what's impressive, and I wasn't going into too much details in the clinical trials, which Rob has already sort of had mentioned, is, in patients who may have had, suboptimal responses to TPO receptor agonists or to other immune drugs. So in the trials have been, been demonstrated that really Phil and Rob is involved in, 40% of patients still respond with their platelet counts with this particular type of therapy. And really the main side effects are really a bit of diarrhea and nausea. So really what this is, I guess, very exciting in that this is probably going to be the next drug that will get approved, hopefully, for ITP in Oz. So that's very exciting. So that's good old Rilza. So next, we're going to go up the chain, right? So, alrighty, what about these bloody jumble of alphabet things? So this is now another family, I guess, of drugs that are trying to basically remove the antibodies from our circulation. And it does this by really exploiting this natural phenomenon that happens in all our bodies. So we actually have, of course, antibodies that are good for us, right? So the whole thing of vaccines and the concept of that is that our bodies it's been exposed to influenza, for example, as the vaccine. So we develop antibodies that will protect us against influenza, right? Now we know that those antibodies one of the ways they sort of hang around for so long in our bodies is they kind of get recycled and have in circulation. To cut the long story short, by understanding that, we're able to basically engineer compounds, one of which is efgartigimod, again, sobriety test, which I just probably failed, that Robert just mentioned, where these medications, as an injection, it's able to basically degrade and stop the recycling of the bad antibodies, right? Because really that's what's happening in ITP patients. Your immune system's confused. It's making antibodies against your platelets. We don't really want those bloody antibodies. So these drugs are able to basically degrade those antibodies so that you're not tagging the platelets anymore. And basically, we one of the first trials, and I think Phil has been involved in this particular trial in Canberra. It's a weekly infusion that's very well tolerated, and it's been shown that 1 out of 5 patients will have a sustained response, to this particular form of therapy as quick as really 1 week after an infusion. Now, I know people look at that and go, 1 out of 5, that's not that great, is it? But again, this is a very heavily pre-treated population. These are ITP patients that really have, low platelet counts despite multiple lines of treatment. And really, I share, me and Phil have many discussions about this, that 1 out of 5 will not be a hell of a lot, but if you're that one patient, though, and we have an option to use this drug, we absolutely would bloody try it, right? So another, I guess, potential drug on the horizon, hopefully. So next, what else do we have? An oldie but a goodie is, of course, targeting these B cells directly, the scouts, right? Or can we somehow disrupt the handshake that's occurring between his buddy, the T cells, which has been very exciting. Now, this ridiculously complicated diagram is basically just showing us the very different sort of receptors. And the way to think about it is this is how the cells engage with each other, okay? And these new drugs basically are able to block that handshake is the way to think about it. And by doing that, it stops that terrible cycle of your immune system keep on telling itself that the platelets are foreign, the platelets are foreign, the platelets are foreign. So these drugs smack us in the back of the head to say, stop it, stop handshaking. They're not bad. And that's what it is. So one of the very exciting, I guess, new agents, it's got a ridiculous name. It's apparently pronounced ianalumab, is that right? Ianalumab. I'm going to call it Iana because that's just easier. So, again, Phil and a number of other different centres in Oz are currently recruiting for this trial right now. And there's other, ways to disrupt that handshake. So I'm not bitter at all, but Novartis rejected me for the Yana trial, so I've taken the other two instead. So there is also, I guess, other agents that we know that kind of does the same thing. So there's povetacicept, which is a subcutaneous injection, and another type of, really small molecule inhibitor with and this breaks convention in that it's got more than three bloody numbers, unpronounceable, but it's another drug that basically tries to disrupt that ongoing immune recognition, basically. So we're really all waiting eagerly, I think, for the results of these trials that are still still recruiting at the moment. Okay. And just schematically show that it's just blocking, I guess, those particular handshake that's occurring between the cells. Okay, so last but not least, we have then, these new generation of drugs that actually has been used a lot in the realm of, I guess, malignant hematology. So there's a particular type of, unfortunately, blood cancer called myeloma, where it involves the cells called plasma cells. Now, the reason I want to mention that is that we've been talking about antibodies. Well, plasma cells is actually the cell that makes the antibodies, right? So once the immune system thinks something is foreign, it kind of then tells the plasma cell, hey, here you go, mate. This is the person that we're trying to kill, which should be influenza or some bacteria, but in our patients with immune disease, it is unfortunately something that's part of our own body, so platelets in this case. So, of course, some very smart people thought, well, hold on a second. So let's just say, we've done all these things. We killed the scouts, doesn't do anything. Why don't we actually directly target the cells that are making the antibodies? And what's really unique about plasma cells is that, I guess they have this very unique marker on them. And I tell people it's kind of like every cell's got a uniform that tells us what it is, right? And I guess you can make, essentially, another antibody that targets that uniform, which is quite elegant. So we're able to kill off just those cells. And this is what this sort of class of drugs are. That is kind of antibodies that we've made synthetically to go kill, I guess, specifically plasma cells in the blood. So daratumumab is, again, an unpronounceable bloody drug. It's a subcutaneous injection where they've just completed enrollment of this actual trial, and really to show, the first sort of two patients in terms of the platelet responses. You could probably see the rise in the platelets that occur within sort of 2 weeks with people getting this particular form of treatment. So, we're all eagerly awaiting the formal results of this particular drug. And in fact, there's going to be now a sort of similar version of, again, unpronounceable name. So this is, in a way, works exactly the same as this daratumumab drug, but it's just a different brand. So instead of a Holden, you're getting a Ford. They're both just as fast. So this trial actually will be starting up hopefully soon in Oz, and I know Phil is involved. Rob, are you involved in this one? No? So myself and Phil will be hopefully we'll be starting this trial soon. And actually, the preliminary results just recently got published last year at the oral presentation in Bangkok. And really, they were just showcasing that 41 patients who received this drug is a weekly injection, just like daratumumab. And you can see that, an average of 4 lines of therapy, so patients have failed a number of different treatments. And really, the quite astonishing thing is, if you look at so the left-hand side, it's the proportion of patients with different sort of dosing levels of this particular drug. The platelet response, so really, platelet count more than 50, or at least 20 over baseline. You can see at the highest dose in yellow, 91% of those patients achieve that. And if you look at that complete response, a platelet count more than 100, I think spine-tinglingly, you could see 82% of patients achieve a platelet count over 100, which really is something that we're eagerly going to see. Is this really real? Is this something that we're going to see in our patients with bigger studies? So really that's going to be, I think, something that's very exciting. And of course, the question is the longevity of that response. And this is just, again, showing the different dose levels over a period of 16 weeks. And keeping in mind, it was only, I think, 8 weeks of treatment. But you can see that out to about 16 weeks, it seems to be that the platelet count is still maintained. But I think the jury's still out. We need bigger studies. We need 41 patients. And I guess that's what this next big study is about, where hopefully we'll see long-term responses for this particular drug. Now, bit of a divergent. Now, I've been talking about really, fancy immune therapies that are going to be, I'm sure, ridiculously expensive. Thank God for the PBS. But I wanted to, change tack a little bit. So this interesting study came out last year by a Chinese group. And what they're doing is actually adding on, sort of boosting the response of drugs that we already have. So eltrombopag, as we know, is a TPO receptor agonist. Now, what basically this study was looking at is, okay, for patients who had eltrombopag and they had 2 weeks of it, but the platelet count, because normally by 2 weeks we expect a rise in the platelets, at the maximal dose is no rise, why don't we add in this thing called diacerin? Now, what diacerin is a drug that's actually mainly used in Asia and Europe as an anti-inflammatory, basically, for osteoarthritis. So we know it blocks one of the kind of hormones that get released in inflammation. And why they thought about this is that in sort of laboratory experiments, they found that the metabolite of this particular anti-inflammatory, in petri dishes and in mouse models, they sort of boosted platelet production. Now, for us, a bit interesting. So they thought, you know what? We give it to people with arthritis, pretty bloody harmless. Let's give it to humans, see what happens. And that's what they did. So they enrolled patients who haven't responded to eltrombopag at the maximal dose, so 75 milligrams, 2 weeks, and want to have a look at the response. And in terms of the patient group, so two sets of the patients, again, had more than 3 lines of therapy, so they failed, other treatments already. And it showed a significant, response. So at 2 weeks with this sort of combination therapy, 13% of patients, that maximal dose, only 13% of an increase, but 44% of the combination had a platelet response. And the response was fairly quick, in about a week's time. And you can see in terms of complete response rates, it was definitely a significant difference in the combination treatment. Now, they didn't make mention, though, when you look at this trial, that the effect was lost over time. So unfortunately, in 12 months, though, only 10% of those patients maintained really that sort of platelet response. And they have noticed the main side effect of this drug has been seen in patients with arthritis with diarrhea. So they do recommend avoiding, anyone at the age of 65. Now, I found this very interesting because this diacerin thing, if you just hop online, and I'm not saying that you should go buy this, but it's actually really quite cheap. So it's about $5 a month. When you buy this drug overseas, and I'm not recommending that. Disclaimer, I'm not recommending that. But it would be interesting to see, whether there'll be more studies of maybe combinations of other type of medications in ITP in the future. And with that, that's the end of my talk, and I'm happy to take questions. Thank you for your attention.
Thank you, Danny. We've got time for a couple of questions.
Hi, Danny. Thanks. That was very interesting. Have the studies looked about the long-term effects on patients taking any of these new drugs? So it's great to get the drug and to help. What are the effects? Thanks.
Yeah. So the short answer is no. The most mature, data will be probably for rilzabrutinib, with my understanding, and Rob and Phil might have helped me out, but it was a 3 year data that I have so far in the Luna, with the Rilza. Longer than that now. So yeah, Rilza will be the one with the longest sort of experience so far in ITP. With all the other drugs, we just don't have enough long-term data. Daratumumab, so that, as I said, that's something that we use in myeloma now for many, many years now, but that's a very different patient population though. They're patients, with really blood cancer. So I don't think we'll kind of extrapolate what happens in those patients or ITP patients, because you're far more immune suppressed in those particular cancer group. Yeah, so unfortunately not. Why they're still ongoing, studies.
I'll just comment on the previous question, if that's okay.
Yeah, of course.
So years ago, drugs were sort of naturally occurring substances that were just kind of almost like randomly tried or observed to work in certain conditions and then developed, whereas I guess now the process is much more that you're identifying abnormal signaling pathways, those complicated slides Danny showed. So drugs are very much designed to target pathways, to block pathways and so on. And if you just take a random drug and see whether it works or not, you're sometimes not going to know whether there are off-target effects. But the way that modern drugs are designed, it's not impossible that there's going to be off-target effects, but it's less likely. The problem is, of course, that these signaling pathways are not unique to things like ITP. They're sort of, found in cells throughout the body in different pathways. So sometimes you can target a pathway in a well-intentioned manner to stop, say, production of antibodies to platelets and so on. But any drug that blocks antibody production to platelets, or like efgartigimod, it doesn't just stop the recycling of antibodies to platelets. It blocks all production of antibodies. And these immunosuppressive pathways, they cause immunosuppression across the board rather than just against ITP. So in a sense, modern drugs are probably, or theoretically, going to be a bit safer in that they're less likely to have off-target side effects. I think the problem is the on-target side effects are going to be potentially more of an issue because these are not unique signaling pathways.
Thank you very much, Doctor.